Good afternoon, everyone. Thank you for joining us. My name is Marissa Waterman. I'm the Marketing Director here at the Academy. Here with us today, we have Dr. Xing Xie, an Assistant Professor at the School of Civil and Environmental Engineering at Georgia Institute of Technology. Today, Dr. Xie will be discussing the recently developed water disinfection approach based on locally enhanced electric field treatment. During the presentation, you'll be able to submit a question by clicking on the Q&A icon on the bottom of your screen. Dr. Xie will be able to answer some of the questions at the end of the presentation. Hello, Dr. Xie, how are you doing? We're ready when you are.
Okay, thanks for the introduction. You guys can hear me clearly, right?
Yep.
Okay, so yeah, so first I just want to mention that I just received a message from the building manager that we are going to have a siren test at noon, but I still don't. Okay, it's coming now. So if you hear some noise background, that's normal. So don't worry about that. Okay, so yeah, thanks for the introduction. Thank you everyone for participating in today's seminar. So so basically we'll go ahead and introduce the LEEFT treatment for disinfection. Okay, so yeah, before everything, before the presentation, basically I want to acknowledge all my students, especially Team 1, and who is graduating semester, and also Jianfen Zhou, who just graduated very recently. Most of the content I'm going to present today are from these two students. I also want to acknowledge all the funding agencies for my work. Okay, so very brief background about disinfection, about bacterial inactivation. So basically, we have a lot of bacteria in our daily lives. A lot of them actually doing beneficial work, but some of them they're still doing, cause significant issues. For example, we have pathogens in water, we have pathogens in food, and also pathogens that forms biofouling. So basically, in a lot of cases, we need to control these bacteria and we want to do disinfection. The most commonly used methods, including chlorination for water disinfection, the heat treatment, pasteurization process for food processing. We also do some biocidal coatings on surfaces for biofouling control. But these are the commonly used methods, but they also have challenges we're facing. They also have problems. So for example, we have disinfection byproducts when we do water disinfection. For the food treatment, if we use a heat treatment, basically they will cause some nutrients and flavor loss. And then for the biocidal chemicals we use for the coatings or for the cleanings, usually a lot of them will cause some environmental hazards. So basically, it's very, very important to, in some cases, to control the microorganisms, and then we have different methods to do that, but a lot of methods we're using now are heavily based on chemicals and that cause problems. So that's why a lot of researchers are still doing, developing alternative methods to do disinfection. So basically what I'm going to introduce today is the LEEFT process. It's also part of this effort. So hopefully, this is still in a very early stage. It's still under, most of them still under institute level development fundamental researches. We're trying to commercialize some of these technologies very soon, but it's compared to all other existing technologies like UV, ozone, they are still in a very early stage. So before we go into the locally enhanced electric field treatment, just give you a short introduction about what is electric field treatment. Okay, so this is the schematic showing that we have one electrode, a positive electrode on the top, and then we have another electrode, negative electrode at the bottom. And then if we have these two parallel electrodes, you will generate a uniform electric field in between these two parallel flat plate electrode. And then the orange lines here indicates the strength and then the direction, right? So uniform distributed. And then what happens is when we have these microorganisms exposed to this electric field treatment, is that we always have ions, like the charged ions in a solution. And then this is a schematic showing the lipid layer, lipid bilayer membrane. So basically, the positive charge ions will move towards the negative electrode side, right? So and then the negative charge ions will move towards the positive electrode. So basically, the movement of these ions will basically squeeze the membrane and generate we call it transmembrane potential across the membrane. So basically, this transmembrane potential will increase when the external electric field strength increase. So at a certain point, when the external electric field is strong enough, and then we will basically the transmembrane potential is strong enough to kind of create a pore or basically break the cell membrane and form some pores on the surface on the surface of the membrane structure. And this process we call it electroporation. So this is actually this process electroporation has been widely used in biomedical field. In most cases, what the biomedical people are trying to do is that they want to control a moderate electric field strength. So basically, when these pores are formed, they can do either gene delivery or drug delivery. Basically, they open the cell membrane and then do some transport some molecules, and then they want the host to be able to reseal or recover. And then those microorganisms, after this electroporation processes, they are still alive. So that's why we can use that for a lot of biomedical applications. The purpose for that is not trying to kill those microorganisms, but what we are trying to do here is that we want to further enhance the electric field and make the pores larger and last longer. And eventually those pores will not be able to recover. And then those microorganisms will be able to inactivate it, basically will kill these microorganisms. So that's the idea coming to my mind. So when I want to explore this process, use it for disinfection. But the real challenge is that to realize a strong enough electric field, even for the normal reversible electroporation process, you need to apply usually several kilovolts of the voltage, because the electric field strength is defined as the voltage divided by the distance between the electrodes, right? So for biomedical applications, usually they use small cuvettes, and then the distance between the electrodes, usually in the millimeter scale, which is already very small, right? And then you still need to apply several kilovolts of voltage. So think about that. And in our case, if we want to use it for water disinfection or food disinfection, basically to inactivate microorganisms, you need to achieve even higher electric field strength. That means you need to either apply higher voltage or reduce the distance between the electrodes. And then, as I mentioned, the distance they typically use is already in a millimeter scale, and then trying to further reduce the distance, there's not too much room to go, and then that will really limit the throughput of the system. So that means we really, there's a big challenge in that to further increase the voltage as well. So because several kilovolts is already very high voltage, and then there's already very high energy consumption, especially when we use it for large scale application. Also, it's not safe for large scale water treatment. So that's why we came up with this idea. Instead of generating a uniform electric field in between two parallel flat electrodes, we try to create a locally enhanced electric field. So by doing that, we are trying to achieve is that we only use a very small voltage, and then with some enhancement enhancement effect, we can achieve electric field strength locally in the system that's strong enough to realize the bacterial inactivation. So there are two strategies we have explored to do this electric field enhancement. One is in the macro scale. So in the macro scale, which means that we can arrange the electrodes in a larger scale. So basically, for example, as we see here, instead of using the two parallel electrodes, we can have one center electrode. So consider this is a tube-shaped reactor. So we have a center electrode, and then we have an outer electrode. Okay, so and then basically based on the theory of the electric field distribution, we can calculate that the electric field strength near the center electrode is actually much stronger than the outer space. So this is the electric field strength is actually reversely proportional to the distance of the point between the point to the center of the tube. So basically, that means that the electric field strength near the center electrode will be much stronger. So it really depends on the ratio of these, the diameter of the center electrode and then the outer electrode, and also the distance would be, within the center electrode. The electric field strength can be enhanced at least, 20 to 30 times easily, at least in that scale. So tens of the about a couple of tens times of enhancement. That's in the macro scale. Just one example in the more like three-dimensional system design, right? So basically, you try to create a uniform size or dimension of the one electrode versus the other. You can basically locally enhance the electric field near the small size of the electrode. And then another scale is a micro scale enhancement. With micro scale enhancement is that if you look at a small feature of the electrode, eventually you will get a very flat surface, right? If you zoom into the center electrode in a very small scale, they will still looks pretty flat. So what we're going to do is we modify the kind of flat surface with some kind of tip structures like nano vials or micro scale vials, basically some like some needles. Really depends on the aspect ratio, means that how sharp these nano vials or tip structures can be. These can further enhance the electric field near the tips of these kind of a one-dimensional features. By doing that, according to the simulation, we can also achieve up to 2 or 3 orders of magnitude enhancement. Really depends on the dimension of these tip structures. So again, so collectively, what we want to achieve is that within the system, we want to apply a very small voltage, and then we still want to realize locally enhanced electric field that's strong enough to do the bacterial inactivation. So based on these two strategies, we have basically proved the concept. So we developed this tubular shape coaxial electrode LEEFT system. So this is a combination of the both macro scale and micro scale enhancement. So as you can see here, so we have this tubular shape electrode, and then we have center electrode, and then we have outer electrode, and then also for the center electrode, we basically modify the surface, and then modify the surface with these tip structures. And then this is a simulation result showing that really when we get close to the center, especially when we have tips modifications, this will further enhance the electric field strength, and then we can easily realize 3 orders of magnitude enhancement. So even though we apply a very small voltage, the electric field strength near these tip structures can still be high enough to realize bacterial inactivation. So this is a prototype of this system. The first prototype. It's kind of just small scale, which we can operate in our on our benches. And then in terms of some kind of details about this fabrication process, so basically the structure is just a glassy carbon, just like a plastic chamber holding the structure, and then you have this copper foil wrapped around. Basically, we can also use copper pipes directly. So that's what we're actually using now. So basically, you have copper electrode outside. So the key component, active component, is really the center electrode. So for the center electrode, we actually what we're using now is a polydopamine coated copper oxide nanovial modified copper. So I just get into some details to explain what it actually is. So we start with a copper vial. Okay, so this is a commercially available copper vial. So it's actually pretty cheap. So for this kind of 1 mile long copper vial roll, it costs less than $10. So it's actually very thin. So the diameter of this copper is thinner than 100 micrometers, about about 80 micrometer. So it's basically very similar to like the thickness of your hairs. So it's pretty thin and pretty long roll, it's kind of very low cost material. So we do one step oxidization process. So basically we heat it up in the atmosphere with certain temperature, and then you will basically grow copper oxide nanowires on top of this copper wire. And then we do another step coating, is that basically this is a polydopamine coating. So polydopamine, so this is a monomer of the dopamine. So it's basically when we do this coating, it's also very straightforward, very simple synthesis process. It's basically we prepare a dopamine solution. You have this chemical, you prepare a solution, and then you just dip your material. In this case, is copper oxide nanowire modified copper. You just put the material into this dopamine solution, and then this dopamine will self-polymerized. So it's also an oxidation process with, certain temperature with oxygen. This dopamine will self-polymerized on the surface of this subject. So basically, eventually you will form a very uniform thin layer of polydopamine on the surface. The purpose of this polydopamine coating is really to enhance the stability or improve the stability of the electrode, make sure it can last for a longer treatment time. Okay, so just give you some images showing what the active electrode eventually looks like. So this is a center electrode. This is a copper oxide modified copper. So right, as I mentioned, it's a little bit less than 100 micrometer. And then if you zoom in and look at the features on the surface, you will see there's a lot of these nanowires on the surface. So showing these kind of tip structures. And then if you use TEM to look at even, enlarge the tips to want to look at more details about the nanowires, you will see the center, the darker part is the copper oxide nanowire, and then the kind of a little bit lighter coating is the polydopamine coating layer outside. You use different methods. You can also quantify what's the exact thickness of this polydopamine coating. Basically, we optimize this coating thickness, trying to basically balance that the performance and also the synthesis time, right? So basically, if the coating layer is too thick, you will be very stable, right? You cover the surface, the electrode will be very stable, last for a very long time. But this is the electric field treatment, so the dopamine coating is actually not conducting. It actually will affect the electric field distribution, and then you eventually, if it's too thick, it's not going to work. So that's why we control we can control the coating layer really by the coating time and also the dopamine concentration in the solution. We can control the thickness fairly well in the nanometer scale. So eventually, I think that the best performance we got is about 50 nanometers. So as you can see here, so this thin coating layer is about 50 nanometers thick. So make sure it doesn't affect the morphology of the nanowire structure that much, but at the same time, it provides enough protection. Just show you the results we achieve. So basically, the most important thing we demonstrate is that we can achieve more than 6 log removal, like 99.9999% of inactivation of some model bacteria using very small voltage, like down to 1 volt, right? So basically, the way you look at this chart is that you have the flow rate, which indicating you can also draw the the curve with retention time, but in this case we just use flow rate. We have log removal efficiency. So basically, if the flow rate is if it is shorter, basically retention treatment time is a bit longer, and then and also the voltage a little bit higher, you will expect higher efficiency, right? So the basic point we want to emphasize is here with a certain level of flow rate, it's about 1 milliliter per minute for our system. We can achieve more than 6. So the dashed line here already indicates there's no live bacteria. So that means it's already higher than 6 log level of removal. So we also test the system with different model bacteria. So most of us tests are still with model bacteria so far. So we test both gram positive, gram negative bacteria. We also test the system with the real river water. We just dose the system with E. coli. But basically, what we try to demonstrate is that they all can achieve pretty high log removal efficiency as long as the treatment time is enough and then the voltage is. All these results are under 1 volt of treatment. We also try to scale up the system a little bit. So as you can see, the 1 milliliter per minute is kind of very small treatment throughput with the first prototype I just showed before. So we try to make longer tubes, right? So in this case is about 10 times longer, a little bit more, about 15 times longer, like a 6 feet system. And then we can also increase the flow rate. So pretty much proportional to the length. A little bit less, right? So you expect that the performance is not as good as the small system, but the flow rate can be increased fairly larger to about 10 milliliter per minute now. Okay, so that's basically the prototypes we have in the lab, and then we try to see, what we have been thinking about what kind of the system can be used in different sections of the water treatment or water disinfection process. So we feel like this can be really applied for all scales. The point of use, a larger point of entry of a building level, or in the primary water treatment. Eventually, hopefully, we can push the technology to that far. But we feel like that's a very unique application scenario is actually in these water distribution systems. So for example, right, because we have this tubular shaped reactor, right? So we feel like maybe we can replace some of the pipelines, right? Not necessarily all of them, but every maybe a couple miles or every certain distance, especially when those places we have the manholes or pump stations, right? So we already have the openings, and then we can potentially replace some of these sections with this tubular shape LEEFT treatment system. So what we can achieve is that we can provide secondary disinfection, right? So I think one of the challenges for disinfection for the current chlorine alternatives is that we can use these UV or ozone in the primary water disinfection water treatment plants to replace chlorine. But in most cases, we still need to have residual chlorine in the water distribution systems to make sure the water is clean when you reach to the end users, right? It will be very difficult to use other process to maintain the distribution power along the distribution system. But in this case, what we are trying to propose, maybe hopefully in the future, is that this LEEFT technology can be a good option for the secondary disinfection. Okay, so theoretically, as I have shown you before, these the LEEFT technology, they can provide disinfection, a very high disinfection efficiency. So theoretically, you just use LEEFT, you can realize a very high performance bacterial inactivation. But we feel like for now the throughput is still very small, and it's still very difficult to completely replace some of the existing technology. So that's why we try to. Oh, sorry, I forgot about one thing about the pipeline systems. Is that we still need to use the LEEFT. We still need to use electricity for to power the LEEFT, right? So because you still need to operate with the electric field. So the best, we feel like the best power source for this LEEFT system is actually just the flowing water. So for example, we can you have this kind of very widely used small water electromagnetic generator. Is kind of you can just use a very small size stream from the pipeline, and then the flowing water. You can just generate enough electricity to power the system. So you actually don't need to use any battery or grid power or any other renewable energy externally. You can just use flowing water in the pipeline system. Okay, so back to the what I just mentioned is basically we feel like LEEFT itself is probably still not very acceptable at this point. It's still a long way to go, but I think LEEFT can be already a good pretreatment technology or can be combined with some existing technologies to achieve synergistic effect, synergistic disinfection. What we have demonstrated here, for example, we can combine LEEFT with ozone. So basically the idea is that you can use LEEFT to treat the bacteria first, and not necessarily to the point that they all get inactivated, but at the point some of them get killed, but a lot of them already get hurt, and then you do the ozonation after that. So ideally, is that you can save the basically the ozonation dosage, right? So the basic mechanism is that if you just do the ozone alone, right? So they need to oxidize, they need to breathe through the cell membrane, and then eventually this can be a distribution curve for the ozonation. So it takes some time. The ozone need to damage basically all the cell structure and eventually into the cellular components. But if you do the LEEFT first, so even though they don't get killed, and then you basically significantly increase the permeability of the cell membrane. So basically, you will significantly increase the diffusion of the ozone into the cells, and then basically make the ozone much more effective in terms of killing the pathogens, killing the microorganisms. Another way to explain this, you can also consider, when we do the LEEFT itself, some of the membranes, right? So you got kind of reversible damages. You have a cell, you have the electroporation happen, so you have the pores form on the cell membrane. So if you don't have a post-LEEFT ozone process, these pores will be able to recover, they will reseal, and then the cells will be fine. But in this case, we combine LEEFT with ozone, and then basically ozonation will basically prevent the recovery of the cells. Anyway, so the mechanism is that really that this is a very synergistic combination of LEEFT and ozone, and then they eventually we can achieve these with some moderate LEEFT treatment, the ozonation dosage can be saved for the same performance, can be saved at least maybe half or even close to 80%. So it really depends on what's the combination, right? How strong the LEEFT is. Another combination we have tried to demonstrate is the LEEFT with copper, because in the previous one you have LEEFT with ozone, you still need to add chemicals, right? So you still, you have the electric system to apply the electric field, and then you still need to add chemicals as ozone. But in this case, with LEEFT and copper, is really we think it's a very nice combination. Is that we can actually generate copper in situ from our system. When we come up with this idea is really from our previously. When we do this, when we do the water disinfection with LEEFT itself, you actually, because the electrode we developed, as you can see, is based on copper, right? So we have the copper film, we have a copper ring electrode, we also have a center electrode which is based on copper. We do notice some copper concentration, we have copper release in the system. So when we try to demonstrate LEEFT, we want to minimize the copper, right? Because if there's any copper in the system, that means that the electrode what we develop is actually not stable, right? So we start with copper concentration about 100, 200 ppb, something like that, and then we eventually, reduce the copper. We try to make the electrode more and more stable. Eventually, the copper concentration is less than 10 ppb, so 10 microgram per liter. So even though we will still have some copper release, but that's what we're trying to demonstrate before. And then, but basically we kind of switch our idea a little bit, and then we feel like, if we can combine LEEFT with copper, we generally release some copper into the system purposely. That can be a good approach as well, right? So in this case, the LEEFT copper system we developed is that we only use one level of electric field strength enhancement. So basically, the center electrode we use here is not the one with nanowires. So it's basically just the copper wire. Okay, so that means that in this case, the copper wire, because it's very low cost, right? It's commercially available, so it's fine that it's not as stable, right? So just basically we can just use that as a as a sacrificing electrode, right? So we just use the center electrode to release the copper, and then basically you will imagine that after a certain period of time, you are running out of your material, and then you can just replace it, right? So anyway, so in this case, what we have demonstrated is that, so again, this is a hydrogen retention time, log removal efficiency. So basically what we have demonstrated is that with, for example, in this case is 1 minute retention time. And then the orange bar here indicates the copper concentration is 200 ppb. And then we realize 6 log removal. Okay, so just give you some background information about copper disinfection is that so usually if you want to achieve similar level, we have the control experiment. You want to achieve the similar level of disinfection. In this case, it's also E. coli. You need to have at least about an order of magnitude more concentrated, higher concentration of copper, about 2 ppm. And then that's also the reason that we usually don't see copper as commonly used as a disinfectant. We have a secondary drinking water standard for copper is 1.3 ppm. So that means that we don't want to have copper in our drinking water more than 1.3 milligram per liter. But the copper disinfection is just like similar to silver, right? So this kind of heavy metal antimicrobial effect. So it has been well known. So it's really because the concentration needs to be significantly higher than silver. So we can see the copper disinfection in some cases, for example, in the hot water lines, in some of these hospitals or hotels, the hot water hot water systems. Some of them they will apply copper ionization cell. It's actually already commercially available system, to make sure you have higher copper it will be higher than the needed for disinfection in the hot water lines to provide enough disinfection effect. Specifically for Legionella diseases, sometimes Legionella will be they prefer this kind of warmer temperature like 30 degrees Celsius, and then easily can survive in the hot water systems. That's why in some of these they have these copper ionization cells. Some people also use the copper disinfectant in swimming pools to replace chlorination, but it's probably not. It's a bit less common. Anyway, so the point here is that usually we don't use copper for drinking water purpose. It's really because the concentration needs to be high enough. But what we want to promote is that if we can combine the LEEFT with copper, right? So similar mechanism, we try to increase the permeability of the cell membrane by using the LEEFT treatment. And then with that, we only need to use a very kind of much lower concentration of copper to realize a very high performance of inactivation. That really, enables the copper to be considered as a candidate for the drinking water treatment. Okay, so and a little bit more mechanisms of the specific systems what we used for the LEEFT is that we have the center electrode, right? So we put the center electrode as a positive electrode. So that's why, when we apply an electric field and also with certain electric current, you can actually release copper, right? So copper is released from the center electrode. It's a copper-based electrode, so copper ions released from the center, and then you basically get higher concentration close to the center, right? So it's similar distribution effect, right? So when it's close to the center electrode, copper concentration is higher. At the same time, we know that most of the bacterial cells, they're actually negatively charged. So when they flow through the system, they actually will, they will get closer and closer to the center electrode. So in the system, we still try to maintain the condition that the cells are not collected by the center electrode. So we do see some collection, but still more than 80%, 90% of the cells still flow through the electrode. Okay, so they were not accumulated, at least most of them are not accumulated inside the system, close to the center electrode. But the point here is that basically the cells will move towards the center electrode where you have the stronger electric field and also higher copper concentration. So this effect really, plays very synergistically to enhance the inactivation performance of the system. Okay, so that's how this system works with the LEEFT and combined with copper. And here's a kind of short video showing that we try to use a TNG system. It's called triboelectric nano generator. So basically, we use triboelectricity. Basically, it's a static electricity to power the LEEFT copper system. So let me show you the short video here. So basically, we have a rotating hand pump. So when this thing is rotating, we generate electricity, and then this electricity is applied to this LEEFT copper prototype to do the water treatment. So we also have a normal, just a water pump, right, connected to the same rotating system. So basically, the idea what we demonstrate here is that we can use this as a small scale point of use water treatment, water disinfection system. We also use another method to power the system is that we can use our cell phone. So we develop a small module and then also a very kind of interface friendly app. So basically you can control the voltage, which means that what's the electric field strength you want to use, and then also the current. So basically that corresponding to how much copper you really want to release to the system. So basically, we also want to use that if you don't have any other external power source and controlling system, you can just use your cell phone to provide a control system for this kind of point of use or point of emergency use systems. Okay, so for another, basically, I just want to talk about the challenges we are still facing. So as we demonstrate, it's a kind of small scale, right? So for this kind of point of use, that's no problem. So we want to push the technology forward for larger scale, for longer uses, right? So one of the biggest challenges is really the electrodes. Okay, so especially basically those with the nanowires. If there's no nanowire modification, right, as you can see, it's already commercially available. Just use the copper wire. That's fine, but we're still in the process trying to study, make sure the copper release is more uniform, right? So it can last longer. But the biggest challenge for pushing the LEEFT technology to be a standalone, high performance disinfection process is really the electrode design for the nanowire modified electrodes. So we start with silver nanowires, copper oxide nanowires, and then now the one I just demonstrated to you is the PDA coated copper oxide. We also change the material from copper oxide to copper phosphide, because copper phosphide is a little bit more stable than copper oxide. But so far we have demonstrated is that the electrode can be used for about 10 days, 2 weeks. Continuous operation, but with synthetic water, basically DI water, very clean water, DI water with model bacteria in a very controlled environment. We apply AC, alternating current voltage rather than DC, and then we also with very small flow rate. So it's still definitely not good enough for a large scale application or for long-term uses, right? For I think much harsher condition can use for a couple hours. That's good enough for some point of use applications, but definitely not good enough for large scale application. So this is one thing we're still working very hard in our lab, and so we try to push the technology forward. But we're not going to talk about the details about the electrode development today. So what I'm going to talk about a little bit more for the rest of the time is that we in the past of the studies we have shown is that we know that we have these systems, right? So it's either we actually have different configuration of the systems. The tubular shape is just one we developed recently, but basically we have these devices. We know that water flow into the device and then water flowing out, and then the system can achieve basically 5 log, 6 log removal. But the system itself still looks like a black box. We actually don't know exactly what happened inside, right? So we never really study what's going on during this LEEFT treatment process. That's why one of our current study we try to figure out is that we want to visualize the LEEFT process. We want to quantify how much contribution is really from the electric field treatment and how much contribution is from other mechanisms potentially exist inside the black box. And also we realize that it's also very important to study the cell transport inside the system, because in our system, the electric field strength is locally enhanced. So there's only very small regions in the system you have very strong electric field. And other places, the electric field strength is just not high enough. So there are different mechanisms that can transport the cells to these regions in the system, but we still need to figure out and quantify those mechanisms. Anyway, so that's what we're trying to do here. We want to visualize the system first. So what we have done here is that basically we move the system onto microfluidic chips. So what you are seeing here is basically a glass slide. Right, so it's a piece of glass, and then you coat the surface with very thin layer of gold. Okay, so it's kind of a standard manufacturing procedure for we need to do it in the clean room. So basically, we can see here you have two bulk coating of the gold that function as just a contacting pad. So this is just 5 millimeters, just a very small piece of the glass, right? So basically these two pads are used for the connection. And then if you zoom in of the gap here, so you will have these nano wedge structures. So the tip is about 200 nanometer, the bottom is about 1 micrometer. So this is pretty much the smallest feature we can fabricate, and at the same time we'll be able to see it very clearly under the optical microscope. We have tried to make smaller ones. In terms of fabrication, you can make much smaller ones, just like those we, in the real device we use. It's actually about 100 nanometer, 100 nanometer or even less in terms of diameter, or 10 micrometers amount of these nanowells. But at least even if we fabricate them, that is durable, but it's very difficult, almost impossible to see under a microscope. And also we, instead of using the nanowells, we have the bottom part a little bit thicker, just because if you have this very thin, uniform diameter nanowells, they will be very easy to peel off from the system. Anyway, so we end up with this design with these nano wedge structures, and then we coat the surface with model bacteria, Staphylococcus, with staining, and then the idea is that we want to do the LEEFT treatment process on this chip. So at the same time, we put it basically under the microscope when we apply the electric field, so we can basically visualize this LEEFT process and then do operando, basically institute investigation of this process. So just show you some results. So this is again the chip we made. We also studied different systems with different of these diameters, but we end up with the optimal design of this chip here. And this is a simulation to make sure we can calculate what's the electric field strength that we can achieve. So we do demonstrate that at least based on a simulation, near these tips, the electric field strength can be several tens of kilovolts per centimeter, which is theoretically required to achieve the irreversible electroporation to kill the bacteria. Here's again the image before the LEEFT treatment. So basically, you will see a kind of all these bacterial cells on the surface. You will see some random spots. They have been stained with red color. Like here is one. There's some others being covered by the words here. Basically, the red color indicates the cell is already dead. So they are dead cells. So basically, the staining is an indication of the integrity of the cell membrane. So if the cell membrane is damaged, and then the stain will be able to penetrate the cell membrane and then the cell will be stained and you will see the red color. So this is before you always have some random, red dots, but most of them are still alive. And then you start the LEEFT treatment. So you can pay attention to the number here. This indicates the time. So now it's still -1 second. And so we haven't started the LEEFT treatment. And then when I click the video, it will start to play, and then you will see the numbers start to become 0 and then positive. That means the 0 is the point we apply the LEEFT treatment. And then you can pay attention to these places, near the tips of these nanowires. So the line here just keep indicating the position of the nano wedges. Okay, so just give you some sign at where those electroporation process will happen. Again, you will see these kind of red dots, which indicates that already died before the electrical treatment. But most of the places you still have a dot area. You have the live cells. Okay, so let's get a video started. So see, now you see the basically when the time gets to about past the 0, and you will start to see some red dots show up, right? So some of them show up here. And then when the time goes by, and then you will see more and more of these bacterial cells near the tips of these nano wedges get inactivated, and then they will get this red color. This is basically we do some image processing. We remove the background noise informations, and then basically that's you will see that those most of these cells, they are at the tips of these nano wedges, they get inactivated. So several information can get here. So first, this process is actually very fast, right? So you can see it's a few seconds. The effective treatment time is actually in the millisecond range. So because when we're doing here, it's not a DC voltage, it's actually electric pulses. So we turn on the voltage and then turn it off for a period of time and then on again. So it's electric pulses. So the effective treatment time is in the millisecond scale. So it's very fast treatment process. And the secondary is that the treatment is really limited to certain ranges, right? So only these places we have strong electric field, you will realize the electric field, the inactivation by the LEEFT. So we use this, we're doing this, basically we have used this platform once we visualize it, we use this platform to do a lot of studies, right? So one of these is, for example, we zoom in. All right, so we're doing the individual cell, individual nanowire. We notice that this is the diffusion of this dye into the cell really start from one point. Like for example, this is 0 second, it's pretty dark inside. Start from 0.1 second, 0.2, like within 1 second, it's become completely red cells, red dots. And then you can see a diffusion of the dye from the point very close to the tip of the nano wedge. The similar phenomenon can also be observed for the negative electrode, right? So this is positive electron, negative electron. This is also a direct support showing that in this system, the damage of this cell membrane is really caused by the electroporation rather than other mechanisms. Because if it's electro oxidation or by ROS, other things, the negative electron, positive electron will be different, right? And also, if it's heat treatment or other chemical treatments, usually you are not going to limit diffusion of the dyes into the cell from just one single point. Okay. So we also do use this platform, as I mentioned, for different studies. So this is basically a visualization of the LEEFT with the different cells and also with the cells that are actually moving. So you can see basically the cells in the solution, and then they will move to the they originally be this in the previous one, all the cells are immobilized, right? In this case, they are suspended in the system. First, we staining with the green dots, and then they move to the side and become activated, and they will change the color to red cells. And then this is basically just like the image showing that after the treatment. So before treatment, they are randomly distributed, suspended in the system, and then you apply the electric field. And after treatment, they are collected on the 2 electrodes, and then eventually they will get activated. So that's why I mentioned the movement of the cells in the system is actually also very important, but it's still we're still doing more study on this direction. Okay, so there's one more thing we noticed in the system, and also feel very exciting about this, is that we achieve very short period of time of the inactivation. We call it nanosecond LEEFT. So as I mentioned before, we already demonstrated it can be very fast, right? A few milliseconds achieve very high performance inactivation. And then we want to push the limit to see how fast we can actually achieve the inactivation. So theoretically, for the conventional electric field treatment, that's the one without these nano wedges, just the flat electrode. Usually, we consider 2 microseconds. Okay, so it's already the limit. So 2000 nanoseconds is 2 microseconds. Because you really need to have enough time for the ions to move and then squeeze the membrane and cause inactivation. And then if you want to shorten that to 200 nanoseconds, the voltage needs to be significantly higher, and then you'll still be able to do that. But if you shorten that to 20 nanoseconds, that's basically the ions are barely move, and then you basically theoretically just cannot achieve any inactivation. We also show that on the chip device, we really observe the phenomenon. At the same time, on the other side is that we notice that with the LEEFT, we actually can push all the way of the treatment time to 20 nanoseconds. So as you can see here, with the 12 volts of voltage, it's about 100% removal, and then even though when the voltage is a little bit smaller, the electric field strength is smaller, you can still achieve fairly significant of microbial inactivation. So yeah, basically we noticed that with the LEEFT system, because the cells are very close to the electrode, and then there's different mechanisms behind, so won't be able to go into a lot of details, but I want to really show you the result is that if this LEEFT process comparing to the conventional electric field treatment, you can either significantly reduce the voltage applied, right? So about 8 times from 55 kilovolt per centimeter, reduce that to only like 7 kilovolt per centimeter. You can see you can even the performance, the inactivation efficiency is even stronger, even higher, or you can significantly reduce the time required for the electric field treatment. So I think that's even more exciting, is that you can, to achieve the similar level of inactivation. In this case, conventional method, you need to apply basically 10 to the 6 numbers of the pulses, with the 20 nanosecond pulse. But in this case, you only need 1 pulse of the 20 nanosecond electric field pulse. You can achieve this even higher, better inactivation. Okay, so this is something we're still we haven't published the paper yet. Still it's still in revision. So hopefully we'll get it published, and then you'll be able to see our detailed results pretty soon. But the why is really promising or very exciting to this, when we feel very exciting to this result, is that as I mentioned, the electric field strength is very limited to certain areas, right? So you want to increase the throughput, you really need to have a very, very good mixing, and you need to try to send all your bacteria or your microorganisms into these regions. So what we can demonstrate here is that if the contact time can be very short, then theoretically our system can still achieve very high throughput, right? So basically you can as long as during the mixing, right, as long as the bacteria touch the surface or touch the electrodes, they will get killed, right? That will be great. So they don't need to stay near they don't need to stay in the regions of the strong electric field for a very long time. So 20 nanosecond is really just, you can even imagine that how short of the time is. Okay, so we use these to do some more, operando mechanism study of the system. For example, we use that to observe the electroporation process. What we can do here is that you can see the purple column indicates that we turn on the treatment, and then the gray parts is the treatment is off. So what we can observe is that when we turn it on, you can see the the field, the dyes diffuse into the cell membrane and then into the cells, penetrate the cell membrane. So we turn it off, it will stop, and then turn it on, you will basically you will get redder and redder, right? And then you see the diffusion of the dye into the cell. So this phenomenon really confirms that in this system is the electroporation rather than other mechanisms again. So because all other mechanisms, you won't, if you use chemical treatment, heat treatment, you won't be able to stop the treatment process well right in the middle, right? So basically what we're doing is we treat it a little bit and stop, and treat it a little bit and stop, and then eventually, the cell membrane disrupted, and then the cell is inactivated. So we see this phenomenon for both the positive electron and negative electron, which really demonstrate that it's the electroporation rather than oxidation or other heat treatment is the mechanism for inactivation. I think this is probably the one last thing I demonstrate here is that because it's the electric field treatment rather than electrochemical treatment. So again, this is really we can consider this is an electrophysical process. It's not an electrochemical process. So that means that these active tip structures or nano wedge structures don't have to connect it to the bulk electrode. So these are the suspended electrodes or the floating electrodes. They are basically have one electrode on the top and the other electrode at the bottom. But these nano wedge structures, they can just be random, not randomly, but they can just align between the two bulky electrodes. You can still achieve the electric field enhancement near these tips. So basically we can pattern different things, and we can achieve these inactivation at these tip structures. We can also increase the density of these tips, and then basically what we're trying to demonstrate here is that in addition to do water disinfection, we can probably use these for antimicrobial surface, right? So as long as we still need to optimize the system, we need to design how it will work the best. But potentially we're saying that this can be a antimicrobial surface application. Okay, so yeah, this is really the last slides. We just want to say that this can be potentially a transformative water disinfection methods. That's what we have been focused on. So a lot of the advantages we have demonstrated here. So very fast process, no disinfection byproducts, and very safe to operate. We also explore the application of this LEEFT for different things like liquid food pasteurization. The liquid food processing or pasteurization is very similar to water, but they're definitely different properties. But we're exporting the potential application along that direction. We have been also collaborating with some collaborators on using that similar process to control the algae bloom. Theoretically, this can also be applied for air disinfection, right? So if you have pathogens in the air, the cleaning process it's also very important. Again, we can use that for antimicrobial surface. So all these applications are still very in a very early stage, but what we are focused on now is one is the mechanism study, and the second is the electrode development for our own group. So, but we're really open to all type of research and developments relevant to this technology. Okay, so just one last slide, sorry, one last slide about take-home message. So disinfection is still very important, and then we still need better technologies for disinfection. With what we're having now is still not perfect. And then last is hopefully what you have heard about today, the LEEFT treatment technology. I convince you that at least can be potentially a very promising candidate for next generation disinfection. Okay, with that, thank you very much. I'm happy to take any questions you have. So
excellent. Thank you very much, Dr. Shea. Now we'll have some time for to answer some questions. If you have a question, just click on the Q&A icon on the bottom of your screen.
Okay, yeah, I can see the questions now. So let me just go through these questions. Okay, so first question, have you considered the possibility of stray current corrosion of adjacent pipes when applying this method? I'm not quite sure about the question, but it looks like you're asking about whether the current applied will enhance the corrosion of the pipes. So I think that to answer this, I think I want to put it that way is that theoretically what we're trying to apply here is the electric field. So we're trying to avoid any electric current eventually. So if you just use a LEEFT, it's just a physical process. So so that's why we prefer to use electropauses. So we use DC currents only for the point of use applications. If in the field you only have cell phone or batteries, it's very difficult to generate electric pulses. You can try to use DC current as well to power the systems for the application, but for large scale applications, theoretically we will just try to use electric pulses a very short period of time, so try to eliminate all the electric current. So lets you avoid any problems in terms of the corrosion of the pipes. Second question, have you considered a possibility? Oh, that's the same question there. Okay, so yeah, the next question, what is the service life of electrodes nano wedges before they need cleaning, renewal, or replacement? How is such maintenance performance? Okay, so yeah, so the devices or the system level devices, I think I already covered a little bit. So the beginning is only a few, 20 minutes, the first generation, and then we already extend the lifetime of the electrodes to, the best condition, more than 10 days in our paper. Then in worst case, at least a few hours should be fine, just for a practical water treatment. But that's for the system level, the nano valves. For the nano wedges, for the chips we use for the mechanism study. So the chips we use is only mainly for mechanism study. Okay, so that we're not going to use that for large scale water treatment, right? So we put that is to really want to visualize the system, to study the system. So that nano wedge is made of gold. Okay, so it's gold nano wedge. So it's very stable under the conditions we applied. So every time once we finish the experiment, we can actually wash it, we can clean it, no problem, and then they can be reused. So the electrode will be very stable, but some of the bacteria, they will probably still stay on the surface, even though we try very hard to clean it. But they can still be reused for a couple of times, and then after a while, we will just use the same chip. We will just use a new chip to make sure we have the good quality of the experiments. But this is a kind of difference to the systems we are really used for large scale water treatment. Let's see, next question. Thank you for presenting. Have you studied interactions of the different microorganisms with the electrode surface? For example, is there interaction attachment, electrical control by fluid, and have their tendency to attach? Yeah. To be honest, very minimum. Dion, thank you for the question. Yeah, so so still very minimum study on that. We only quantify that most of the cells will when we measure the cell density in the effluent, most of the cells were either in the effluent. So, but we do see some accumulation in the system. But really, because the system was not operated for very long, especially for most of the experiments, so we don't see a significant impact of the accumulation of the fouling of the system to affect the performance of the system. So I think for long-term applications, especially in real case, theoretically we can control the direction of the electric field. So if basically we can reverse the electric, just like the backwash process. If we see accumulation of the cells on certain electrode, we can reverse the electric field, try to have some repulsion forces to clean up system, or even we can purposely generate some electric current to clean the surface. But that's still a lot of things to do. That's, and to be honest, just we haven't done much on that. Okay, let's move on to the next. Is this technology proposed for use in potable water treatment? If so, would the resulting copper ions resulting in the impact meeting the? Yeah, so at least for now, we are focused on the potable water treatment, right? For drinking water treatments, at least the water source we're going to treat is the most cleanest, right? So comparing to wastewater treatment or liquid food treatment. So liquid food treatment is even much more complicated or difficult to treat. For drinking water treatment purpose, we're actually not worried too much about the copper release. So at least we were trying to minimize the copper release, right? So even though we have some, we will be, even if in some cases, if we come to combine the LEEFT with copper, we will control the copper release. With smaller amount of copper, copper is actually a essential elements for our health. So I think you can have at least up to I think 2 milligrams of copper, if I remember correct, 2 milligrams of copper per day. But usually you will get it, enough from your diet. But if you can control the copper concentration within a couple hundred ppb, and then with the water you drink, there shouldn't be any issues for your health, and there shouldn't be any problem meeting the regulation. I think this is probably the last question here. Is disinfecting wastewater could be these needles develop, grows, and recover? Yeah, so kind of similar questions to one of the before. This is really in the very early stage. I want to admit that. So really depends on what kind of systems you want to use and what kind of application you are. Whether in some cases you probably want to only use a larger scale, macro scale enhancement without the nanowells. With the nanowells involved, the electrode will be much more fragile, right? So, but the large scale design in terms of using the concept of the non-uniform electric field, locally enhanced electric field, to do some water treatment relevant processes. I think that's at least something looks very promising to me. At least we will quickly demonstrate some practical applications with this type of technology. I think I do the question in a reverse order. Okay, so let me see some new questions coming up. Can you tell us the issues of inorganic fouling in real water treatment application? In some conditions, they may not be able to control, especially for application large flows. Yeah, so in one of the studies, we do notice some inorganic fouling on the surface, but again, it's just too early for me to give you a good answers on that. That's definitely something we need to consider for large scale applications. But again, there will be mechanisms to study how we control it. When it's not really controllable, how can we clean the surface, right? What is the system resistance to chlorine? Does it degrade electrode more quickly? So really depends on the electrode used. One of the recent publications is not from our group, but from one of the students who was a visiting student in my group. He collaborated with some other people, and then I think he demonstrated a system with the LEEFT with chlorine. So what I talk about today is the LEEFT with ozone, but there's a recent ESNT paper about LEEFT, and I think he just used electroporation process with chlorination. It's a combination of that. So again, the durability of the electrode is really one of the key to make this technology work. But for a short-term demonstration, there's no problem. But of course, if the system is more corrosive, right? So basically, we're trying to study the more like fundamental process of what will affect the durability of the electrodes. Pretty much there will be two directions. One is the mechanical property, how when you have flush water, right? So it's the mechanical flushing. And second is the basic electrochemical condition, right? So both the potential will apply, and also the basically the corrosive of the water, right? The pH of the water will affect the durability. We're also studying the coating. How can this coating be able to protect the surface and then at the same time create a minimum effect to the electric field distribution, to the electric field treatment performance. How about the cost comparison? How much LEEFT will cost per liter of water? Also the cost, it's hard, still very early stage. It's hard to really provide a very reliable cost analysis. We do some analysis for some of our systems. So for example, in terms of energy consumption, so the conventional LEEFT, usually people use about 10 to the at least 10 to the 4th or 5th joule per liter of liquid food treated. So, but that's usually for liquid food. But some of the studies also use water as a synthetic food for the conventional LEEFT system. So it's really kind of energy intensive. That's why in the food industry, the electric field treatment is actually already existing for several decades, but still not largely implemented for large scale practical application. It's really because the energy consumption is still too high, and also that when the voltage is high, sometimes they will cause some issues of the system. It's not that easy to apply. But anyway, so with the LEEFT process, in our small scale demonstration, we can reduce the energy consumption to only a few joules per liter of water treated. But again, this is in the very controlled environment, very small scale. For the LEEFT copper, the combined LEEFT and copper system, we also do some estimation of the cost of the water traded. I think the number we report is about $0.1 per cubic meter water traded. So it's very low cost in terms of the treatment cost. And then most of the treatment is actually in that case is from the copper. So it's not from the electricity we apply. So it's very small amount of electricity you need it for the LEEFT copper system. But you still do need about, like, in our case, the ideal case is 200 ppb of copper in the system, right? So you can just estimate how much copper you release in system, multiply it by the price of copper in the market to get a price. No, in this case, we just consider the consumable parts for the operation. Right? Have you tested Jardier instead of no? So we haven't tested any protozoa. So we are very interested in doing that, but we just don't have the capability of doing that. There's some studies about protozoa like the Cryptosporidium and then Jardier, like back to 1990s. I think it's Professor Charles Haas. He combined the electric field treatment. So at that time, it's just a conventional electric field treatment with help of the current to see whether this electric field treatment will increase the performance of the chlorination for this protozoa inactivation. There's some papers back to then, but basically the general conclusion is that the electric field treatment itself doesn't work that well. So even though the treatment efficiency is still less than I think 1 log, something like that. But if you can combine that with chlorination, that can at least improve the performance of the chlorination for the protozoa inactivation. Okay, so next one, it sounds like this technique can also help apply at household level at point of disinfection. Yes, yeah. So we always try to start with small scales, right? So, but our vision is that potentially this technology can be applied for much larger scale. So, but we start working with point of use devices, with small prototypes. But we feel like the ideal case is that it's probably not good for portable devices. Basically, if you want to carry those devices for in the remote areas, you will still need to have this electronic unit, right? So we feel like the best case is that still either in a household or in a building, you have the places to install this electric part of the system. So in that case, you can still you will be able to use pulse treatment, and you can control the system, how much electric field use, or in other cases maybe you can combine it with some copper or other things if you like, but you can have a very well controlled system. I mean to ask compared to disinfected by chlorine in terms of the cost, or yeah, I think at least it will be comparable to chlorine or even less. That's theoretical estimation. One thing I can emphasize on this in terms of the cost is that for all these conventional methods, right, either chlorine or UV or ozone, right? So basically we are treating the water as a bulk, right? So we're not just treating the pathogens or bacteria in it. We're treating all these water molecules or everything in the water, right? So that's why you actually doing a lot of things actually which is actually not necessary. So with the LEEFT technology, the strategy is that we only have very small areas. You have this locally enhanced electric field, and then what we try to do is in the system, the microorganisms will be transported to those agents by different mechanisms. So something we already know, but some we already is already under under study. But basically, either by mechanical ways, right? You have mechanical mixing, or it's like electro using the electric field, basically. And then basically, we're trying to treat it's just the microorganisms themselves. We're not treating other parts of the other components in water. So that's why theoretically, this can be a really energy efficient process. Okay, so comparing to a lot of existing methods. So, but there's still, this technology is still very early stage. There's still a lot of things to improve. Is electrical disinfection being considered by a public health agency for water treatment? So in the food industry, the electric field treatment, the pulse electric field treatment is I think it's already been certificated, or at least being used. So they have the really commercially available units which can treat hundreds or thousands of liters of liquid food. So at least it's been considered as a acceptable food pasteurization technique in the market. I don't think there's any large scale large enough studies for water treatment. So that I don't think that's if you want to meet any regulations or standards, I don't think there's anything available there yet. Next question, please address the fouling of the tubes, or also can the system be recycled after cleaning? I think I already talked about that a little bit. So yeah, the chips we reuse it, but for the system we didn't try to. So basically, when the nanowires, we will see the basically during operation. For this device system level, you will see the nanowires getting shorter and shorter. I forgot to put the SEM image here, but basically the nanowires get corroded, and then shorter and shorter, or some of the nanowires get directly peeled off the system. So after a certain time, when you reach the end of its lifetime, you basically you don't see much nanowires remaining on the surface. So it's not sort of the fouling is really still not a significant concern at this point. It's really more like the durability of the nanowire. For liquid food disinfection, the cell lysing may take place on food cells. Does this affect it? Yes. Yeah. Less has been research. Yeah, less also been studied in the food industry. So that's why it really depends on what kind of food they treat it. I think most of the food is already in a liquid form. Okay, for example, apple juice, right? So it's already, they don't have the cell structure yet, remaining in the system. So this treatment is actually in the food industry, not only for pasteurization. Another application is used to enhance the extraction of components in the food cells, or for apple juice, probably not apple juice, but for some other juice extraction. So you use that to treat, I think for, I don't remember, but basically for you use that to treat the solid foods, so they can have the solid foods exposed to electric field, and then after that you do the normal squeezing or mechanical extraction process, so that will be achieve better performance. I think if you Google food processing electric field treatment, something like that, you will also easily find studies that use that to treat potatoes. So I think one study showing that you provide better performance when you do the potato chips, and then you do the electric field treatment, you will make the chips more crispy or something. I don't remember exactly, but you can do some Google search or find some cool videos about the electric field treatment in the food industry.
Excellent. Thank you so much. I think that was our last question.
Yes.
Thank you everyone for your questions, and thank you, Dr. Sheh, for sharing your knowledge with us today. If anyone would like to reach out to Dr. Sheh directly, please feel free. If you could just type in your email address in the chat for everyone, that would be great. We now have an added feature to our webinars. It's an after party. So Dr. Sheh has agreed to stick around for a few minutes to answer any additional questions you might have. We will end our event and stop recording, but if you want to stay on, you'll have the opportunity to ask some questions in a smaller group, and you can even unmute and ask your question if you'd like to. A few items to mention before we wrap up. We have several webinars planned in the near future, so you can go to aaees.org/events to check them out and register. If you're interested in sponsoring an upcoming webinar, please reach out to me directly at mwaterman@aaees.org. And just a reminder, if you're not yet an AAES member and you're considering joining the Academy, please email me and we can discuss our membership options. Last but not least, our PDH certificates for this event will be sent out within the next week. And that's all for today. Again, if you'd like to stay for the after party, please do. Thank you so much and have a great day.
Thank you, everyone.